Okay, today the last section we are going to discuss about this auto analyzer. Okay, auto analyzer. So in this auto analyzer, it is mainly used to measure the blood chemistry and displays this on a graphical readout. So we are just um, measuring the blood chemistry, and that is and it is displaying. Okay. So this is the block diagram of this auto analyzer. Here in this we are first there is a sampler. Okay, this sampler is samples are feeded in this sampler. Okay, samples are feeded in the auto analyzer by using a sampler. Here in this sampler we can able to place 128 samples. Okay, 128 samples at a time. So the sampler contains the holes in which the sample tubes can be placed. Okay, the sample tubes can be placed in this holes provided here. Then this is passed to the mixing and proportional circuit. This mixing and proportional circuit is the heart of the auto analyzer. So the proportionality pump is basically a simple pedestable pump working simultaneously. This is the pump. This works simultaneously. Okay, it will take the first samples given to the mixing or proportional pump, then take the second samples and so on. Okay, so in this mixing or proportional pump, it mixes the reagent. Okay, then there will be a cleaner. Okay, so after the first sample is mixing with the reagent, it will pass through this tube. Okay, after passing the first reagent and sample, there will be some air bubbles or gap. Okay, the air bubbles, then the cleaner will pass. Okay, the cleaner will pass through the tube. Then after the cleaner also there will be some air bubble. So first samples and reagent will pass through the tube, then there will be some air bubbles, then what else? Cleaner, cleaning the tube, then cleaner, then air bubble. This process will continue on as the number of samples will pass through this uh, samples with reagent, then air bubbles, then cleaner, then air bubbles, then again the next sample and the reagent, then like that it will goes on to this. Okay, so the next after this it will pass on to the dialyzer. Okay, it separate the interfacing solution from the sample by permitting selective passage of the sample through a semi-permeable membrane. Okay, in this dialyzer it separates the interfacing substances from the sample material, separates the interfering substance from this material by permitting selective passage of the sample components through the semi-permeable membrane. Here it will allow only the selected membranes, selected sample components which is passed through the semi-permeable membrane. Okay, then it pass on to the heating bath. Okay, it is used to heat the fluid continuously by to the temperature of 37 degrees Celsius. Okay, to exact temperature here, in this heating bath, this fluid, that is here the fluid is mixing with the reagent, then it is passed through the dialyzer. Here in this dialyzer, this particular samples alone will be passed through the semi-permeable membrane, then it pass on to the heating bath. In the here it heats the fluid continuously to the exact temperature. The temperature is critical for the color development. For identifying the color or developing the color, this temperature is important. Then again is passed on to the colorimeter. Okay, so this colorimeter is used to measure or monitor the change in the optical density of the fluid which flows through the tubular flow cell. So the color intensity is proportional to the substance concentration and is converted into the corresponding electrical voltage. Then again it passes on to the graphical recorder. It is used to convert the electrical signal into the graphical display. Okay, so here hope you understand from this sample it pass on to the mixing and proportional circuit. It was mixed with the reagent and then passed to the dialyzer. It allows only a particular samples by passing after passing through a semi-permeable membrane, then to the heating bath. There it is heated for a particular temperature to get the color samples, then to the colorimeter, then to the graphical recorder. So here we have to consider, there will be some points we have to consider, that is sterilization is needed for the samples and glass tubes. So this is passing through the glass tube, so sterilization is necessary. So that is after, if there is no sterilization means if one samples have diseases means other samples also shows the diseases if the sterilization is not done properly. So the sterilization is necessary for this in this auto analyzer. Okay. So the sampler aspirates samples, standard and wash solution to the auto analyzer system. Proportionating pump and manifold. It introduces or mixes samples with the reagent to the effect the proper chemical color reaction to be read by the colorimeter. It also pumps the fluid at precise flow rates to other modules as proper color development depends on the reaction time and temperature. Then dialyzer. It separates the interfering substances from the sample material by permitting the selective passage of the sample components through a semi-permeable membrane. Then heating bath. It heats the fluids continuously to exact temperature, and the temperature is critical to color development. Then the colorimeter. It monitors the change in the optical density of the fluid stream flowing through a tubular flow cell. Color intensity is proportional to the substance's concentrations are converted to the equivalent electrical voltages. The recorder converts it converts the optical density electrical signal from the colorimeter into a graphical display on a moving chart. Okay, that's all. Thank you.